This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 771, recorded on June 18, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. It's a gorgeous day. Another in a series of gorgeous days here in Western Mass. It's 81 Fahrenheit, 27 C, humidity 31%, and clear skies. Just absolutely lovely.
Very similar here. 28 C, which is 82 F, and sunny. Lovely. Yeah. Also joining us from New York, Amy Rosenfeld.
Hello.
I guess your temperature is the same as mine. Your weather, that is, right?
I'm in the tundra.
You're in the tundra.
We have this lovely vent. It propels some air very violently. Then you get
the climate control system in the Hammer building. I remember it well. You could be in completely different weather zones at this point.
Yeah,
you could even be completely different weather zones in different.
I have to say though,
it's quite. I'm just waiting for this.
In response to COVID, they did turn up the air circulation in Hammer, the building we were in.
Right.
And now there's an audible whoosh coming out, which never happened before, at least in my office. Saul always used to complain that there was a windstorm coming out of his ceiling, but I never had that.
But that was mostly coming out of Saul.
No, that was hot air.
It was very blustery in Saul's office. It had nothing to do with the air conditioning.
So somewhere in this building, there's a switch that can turn the air up. They always used to say, we can't do anything about it, but apparently they found
it cost them money. That was the problem.
Is this the smallest TWiV? Has there been a two TWiV? I guess there have been two TWiVs.
We had some two-person TWiV. Well, TWiV 1.
Well, you've had beginning
that's right.
with two people.
Dixon and I did
first
too.
One was just Dixon.
That's correct. That's correct. And I think recently we've had, not in the past year maybe, but past few years we've had some twos. For the American Society for Virology, a few announcements. If you would like to learn more about vaccines, you can go to a town hall where there are two experts. It's not really a hall. It's just a Zoom session, but they call them vaccine town halls. You get two experts. You can ask them questions. And sometimes it's just one or two people there, and sometimes it's more. But they're all very nice, friendly people, no intimidation. You can sign up at asv.org/education. Or if you search for ASV Town Hall, you will get there. And some of them are now in Spanish.
Excellent.
There's one on June 22.
And these are actual virologists, not just some random schmo you found on Google. So it has the benefit of useful information about vaccines.
As we said last time on TWiV, someone said, how good are you? I wanted to know, why can you talk about viruses? And for those of you attending ASV, the meeting this year is online. You have to register for it. For the social events in the evening, we're looking for people to host themed breakout rooms, as described in the recent email to members. For example, so far there are two themed rooms that the plant virologists are planning. I hope they have plants behind them.
Yes.
At least one lab alumni group will have a theme room. Another is planned for teachers of undergraduate virology. You can contact ASV at asv.org if you have a theme to suggest and publicize. Okay. And TWiV is going to be Friday at 5 p.m. That's the 23rd of July. And I believe it's going to be live streamed. Cool. I know it's going to be live streamed for the members. And they told me, just give us your live stream link, which means, I can live stream this thing that we're on now.
To YouTube, yeah.
To YouTube. And so I will give it to them. And then other people can watch on YouTube also.
Great.
Which will be fun. Right, Amy? We love live streams, don't we?
Yeah. So the live TWiV at ASV would then be open to the public.
Yeah, it will be open to the public. A few people have asked, and I thought that it wouldn't be, but since they're leaving it up to me to provide the live stream, it will be open. And it's fine. It's the last event of the meeting. It's Friday, 5 p.m. We will have billions of people watching. Well, maybe not billions.
Maybe not billions.
A few hundred.
Well, I mean, Alan's going to tweet it out to all his friends.
All 10 subscribers.
The card flipping people.
Yeah. Oh, yes.
Right.
Yes, absolutely. I've already promoted the show to
The card.
to the
card
local
game.
card group.
All right, we have 2 papers for you today, a non-COVID and a COVID, so that we ease you back into the rest of the virology world. And
There's other world of viruses.
It's quite fascinating, actually.
This
And
is a
diverse
really
and
cool
interesting.
paper.
And this is one of...
It's computational.
It's okay.
I am not that interested in computational
I'm
biology.
sorry.
It's okay. Interested isn't the right word. It's
Excited by it. You're not too excited.
You can make it say whatever you want.
Well,
and there's no way
I'm
to
not
disprove
well versed in it, but this really demonstrates the potential of what you can do with it, and I'm very impressed.
How can you not?
Yeah, but you have to be really careful.
You do.
Of course.
You can make it do what, and you can make it say what anything.
Right,
anything
garbage
you
in,
want.
garbage out.
I mean,
But if you have a paper by a good group that you're putting some trust in the peer reviewers, and you can do some of your own analysis. You can read the paper and see if what they're saying makes sense, which it does.
And hopefully it gets good peer review, right,
Yes.
by the appropriate people. Like, I could not peer review this paper.
No, I couldn't. No.
I would not have the qualifications, but, Eddie Holmes or his ilk would, or her ilk would, Anyway, how can you not like a paper where they talk about sharp belly fish and carpet pythons?
Yes.
I've never heard of a carpet python or a sharp belly fish. But this paper, PNAS, 100 million year history of bornavirus infections hidden in vertebrate genomes, from a group of investigators at Kyoto University, Kawasaki, Kojima, Mukai, Tomonaga, and Hori. And let's talk a little bit about bornaviruses first. And by the way, this is probably not open access, right, Alan?
No, it is not open access.
Yeah, back to the non-COVID world.
Yeah.
And there's
non-COVID papers were were always not open access.
They were. It was spotty
were.
depending on journal, but in PNAS, in closed access.
There's also a very nice commentary accompanying the article by Robert Gifford.
The commentary is open access. Strangely
that's
enough.
interesting.
Yeah. I downloaded the commentary and then I went to download the paper and it wasn't giving me the PDF. I said, I actually have to log in for the paper. But
yeah, the commentary is open. That's good. He's from the University of Glasgow, and we actually discussed his work previously on TWiV. This interesting story of how malaria studies led to the introduction of a retrovirus into birds so long ago, I don't remember. It was fascinating. Anyway, his commentary mapping the evolution of bornaviruses across geological timescales. So first, bornaviruses. What is a bornavirus? It's we actually had a TWiV called Borna That Way.
Yes.
That Way.
Borna That Way, yeah.
Isn't it? Is the song Borna This Way or Borna That Way?
I think it's Born This Way is the song.
Oh, because the TWiV was Borna This Way. Oh, you're right. You're absolutely right. I am wrong again, as usual. Borna disease virus, BDV, infects a variety of mammals, and it causes a neurological disease in horses.
Sad horse
disease, which is poor. I mean, why the long face?
Right.
I have bornavirus disease.
No, because I'm a horse,
Borna disease or sad horse. And here's an interesting one. The name Borna derives from an 1895 outbreak that occurred near the town of Borna in Saxony, Germany. Folks, we're naming viruses after places. Well, it was 1895, which is at the very beginning of our virology.
But that doesn't make it right today.
I don't mind naming things after places. I understand the objection.
There have been a couple of other suggestions that came through that maybe we'll get through in the letters. That I actually came up with one independently that I think is in one of the letters as well. We could do like hurricanes and give them people's names.
I think that they wrote about that. About why they didn't give people's names because they came.
They want to be neutral.
They decided that they decided that there were not enough or something or other. I forget.
There are not enough people's names.
But
how many Greek letters are there? 26.
Whatever. Move on. But the fact is that what we did in history doesn't make it right today.
Yeah.
We had
We have
slaves in
Juneteenth
history.
coming
Doesn't
up to
make it
remind
right.
us of, yes.
Right. We had slaves in history. So I can read, you know, Tom Sawyer, all about the Negro and the slave. Doesn't make it
I don't
right.
think that there's a comparison between slavery and
Well,
naming
no, right.
viruses
That's a
after
false
places.
equivalence. But the point is, just because
It's
of
a little- it's not a
just
false
because of
equivalence.
precedent doesn't mean that we have to stick with something.
Okay. Anyway, this
Right.
one is called.
It's not a false equivalence. It's
It's
just
an
an
exaggeration,
exaggeration
yes.
to make a point.
This is Borna disease virus, and the name is sticking. We're not going to call it Delta virus.
The
but
people
apparently
in
this,
Borna are not being beaten up for the fact that this sad horse disease came from there. But it did.
Right.
The outbreak was, well, it didn't even come from there. I mean, it was discovered there because there was an outbreak in the horses that, as the review article on this says, it decimated the Prussian cavalry that year.
You took away 10% of them, you think?
I don't know if it took away 10%, but maybe it took away 90% of them.
Now, for many years, the people have been trying to make a connection between Borna virus infection in humans and human mental health diseases like schizophrenia and depression. And in fact, our good friend Ian Lipkin worked on this for many years. But I don't think there is a connection. What has recently been discovered, and we talked about this on two other TWiVs.
Yes.
We talked about one, so there are these 13-lined squirrels that have Borna viruses, and sometimes people get them. And I did a TWiV in Rotterdam with a guy who works on this also. There was a recent zoonotic event in Germany. 3 men died of encephalitis. They were all breeders of exotic squirrels. And eventually they figured out that the 3 men all got variegated squirrel bornavirus 1 from their precious squirrels. This virus apparently was introduced into the German captive squirrel population. Who knew there were captive squirrels?
This was incredibly educational for me. I hadn't the foggiest idea that there were squirrel fanciers. But okay, yeah, you know.
And by the exotic pet trade and in 2003, and has caused 4 people at least to die. So now the idea is that, okay, so horses get this infection. I think the fellow in Germany told me that these horses probably get it from rodents that are in the stalls. And that people probably don't have for schizophrenia and depression, but they do get encephalitis from it. So that's Borna virus, a negative stranded RNA virus.
And I have to say, it's a wonderful coincidence that Borna has RNA in it.
Look at that, yeah.
Right. I mean, we have picornaviruses, which are not named after the town of Picorna.
Yeah.
But yeah.
If you live in Saxony, you have RNA at your core. If you live in Borna in Saxony, that is. Now, so that's the virus. And what is the paper? Now, the paper is all about Eve. All about Eve.
All about Eve.
Wasn't that a play or a book or something like that?
I think both.
Eve is endogenous viral element. And the discovery of these has made it possible for us to figure out how long ago viruses were around. And as you saw from the title, in this case, 100 million years ago, and maybe even earlier, of course, but we can pretty much say, and you'll see as we go through this paper. But these are viral sequences. They're DNA sequences in our germline. So they happen in many species, not just humans, not just mammals. And what happens is whether it's a virus, an RNA or DNA virus, little pieces of the genome, in the case of a DNA virus, they can just get into the nucleus and get integrated. And sometimes they can be integrated into the germline, so then the species passes them on to offspring. But in the case of an RNA virus, we have reverse transcriptase in our cells in the form of line elements and others.
Which comes from previous viral infections.
They come from previous viral infections. And these reverse transcriptases can copy RNA, and the DNA can get integrated, caused by this line element. So these are random integrations that have no functional consequence, although some people think they might. They're just very small pieces of viral genomes. But the beauty is you can study the evolution of viruses because they're in the germline, so they're passed down, and you can see where they are in the chromosome in different species, and you can use that to calculate a timeline, essentially.
Right.
And so that's
and then
all
based on the sequence divergence between different eaves in different species versus the sequence divergence within those species, you can even, as in this paper, trace back and say, well, this is most likely a virus that infected the ancestor of both of these species and then evolved with them, or this other one might be a virus that infected both species independently after they had diverged.
Right. So the age calibrations are done based on finding orthologous eaves in related species.
Right.
And you can see when it appears and data, because you have to also know the phylogeny of all these species as well.
Yeah, so this is a heavily computational paper, but there's also a tremendous amount of virological knowledge going into it. So they're using the computer to do the data intensive parts and mine the sequences, because there are gazillions of these. That's an exact number. And then they're looking at them and saying, okay, well, what makes sense? The algorithm highlighted these as possible similarities, and then we're going to put them manually into some kind of a sensible plan.
So to start, what they do is, so they're going to look at EBLs, which are endogenous Borna-like elements. Okay, they take all those Borna-like sequences in the genomic data of eukaryotic species, right? So there are lots of genomes sequenced. You can go search them yourself. 969 eukaryotic species. They just search for Borna-like sequences. So you have Borna genome sequences, and you say, is there anything similar in the genome? And the Borna virus, by the way, the genome encodes 6 proteins: nucleoprotein, phosphoprotein, matrix, glycoprotein, the envelope, the RNA polymerase, and the accessory protein X. And others have previously found endogenous elements from the N, M, G, and L mRNAs. And they found they found all Borna-like elements, 1,465 of them in 131 vertebrate species for the various viral proteins. So you can get DNA copies for these individual messenger RNAs integrated. And then they said they can classify these to try and figure out which. There are a number of different Borna viruses, so they try and figure out which these came from. And they can find. So the problem is, you're searching with contemporary sequences, so you're only going to find things that are related to it. And as they say later, there may be distantly related Bornas. We're not going to pick that up. So they end up saying we found numerous previously undetected loci, and so now we have a big database of these elements. So then they take all these elements and they do the integration time based on gene orthology. So the endogenous element in different species.
Right.
So and that's where the very heavy computational biology comes in, and that's where they can say we could trace bornavirus infections back to 100 million years ago. So the oldest records were previously reported in Afrotherians. I think that's elephants, right?
Yeah, so I looked these up, and Tethytheria is
83 million years ago. And they found 6 elements that were orthologous among Boreutheria, suggesting that the oldest Borna infection occurred at least 96.5 million years ago.
Right. So Afrotheria are it's a bunch of different things: some moles and shrews and aardvarks, and also elephants and sea cows.
Okay. So we also found 18 integration events in the Mesozoic era, which occurred in the ancestors of Afrotheria, Tethytheria, Metatheria, primates, and Rodentia.
That's a long time ago.
I also found an integration in ancestral passeriform birds during the Mesozoic, which would be between 66 and 82 million years ago. So multiple vertebrate lineages in the Mesozoic era already. And they also find that the ancient Bornas infected more vertebrates than we know today, right? So modern infections with Bornas, ungulates, shrews, squirrels, humans, birds, garter snakes, but they found mice, which aren't known to be infected today, Afrotherians, marsupials. So you can find, this tells you that there's dynamic infection going on, or maybe, as they say, we just don't know how many species are infected today.
Well, and because of the extinction rate, there are a lot of species that they're not going to be able to measure.
That's right.
So they're looking at these sequences from extant species and piecing together a tree that's got missing members to it.
Yeah. So they have infected more lineage than we know of today, and our understanding of the extant Borna host range may still be insufficient. Then they also looked at the geographic distribution of these ancient infections. Where on the Earth did they happen, which is quite interesting. They happened on different continents.
Yeah, but the continents were joined.
Mesozoic.
In the Mesozoic?
I thought that they were.
Well, there's Laurasia, so that's a different.
I thought that parts of the continents were
just looking
some
up
of them.
I could be wrong.
Laurasia and Africa in the Mesozoic era, Antarctica or Australia around Cretaceous, Paleogene boundary, and possibly Eurasia, Africa, South America in the Cenozoic era. So that's pretty neat that you can go to that. You can assign geographical locations.
I'm trying to find a good Mesozoic map.
Mesozoic map.
My
of
road
Earth.
atlas only dates to the late Cenozoic.
And they found bornavirus in.
Oh yeah. So Laurasia and Gondwana in the Mesozoic. So Pangaea was breaking up and separating into continents. So yeah. I mean, Amy's point is good. A lot of some of the continents that we now identify as separate were in fact joined into these megacontinents in the Mesozoic.
So that means that animals could move around and
they could move more easily between the continents
and perhaps take their viruses with them.
Yes.
And
I think that's I think that's also like. I think when you had Simon on for TWiV and TWiV, you talked about like the bat populations, and he talked about like how you find the same bat populations like on the Gold Coast as on the bump in South America because it fits into each other. And then, So, I mean, I'm not really good at, you kind of need Dixon.
He was around for all this.
Well, I wasn't going that far.
Sorry, I shouldn't do that when he's not even on the show.
I wasn't going that far. I was referring to his, his interest
the Mesozoic. Yes, I was in high school at the time.
They also say that in primates, they seem to have been endogenized with multiple kinds of bornaviruses rather than a single lineage repeatedly endogenizing. And during each different era in primate evolution, there would seem to be different bornaviral lineages, present, which you can track by this analysis. And they say the genetic diversity in these ancient bornaviral lineages is greater than we see today. Which they say maybe we had more infections, different species back then, and maybe in part because of these land bridges, more species could get together and so forth.
Well, and there may have been better reservoirs that are now extinct.
Yeah.
Or there might have been just more interactions. Like we don't really live with our cows anymore, right?
Apparently,
our milk cows.
Yeah, even Daniel doesn't.
On a separate farm.
Daniel has 2 cows in Uganda. He misses them.
Oh.
He wants to visit.
It wasn't really
That must be really inconvenient for the milking.
Well, I think he's paying for their support, right?
He's paying for their support. I see.
People who...
I was going to say, I think he does.
But he knows the names and he goes to see them, right?
More of an absentee gentleman farmer type of thing.
Yeah. But we don't really live with the farm animals as we used to.
They're quite separate, yeah. Although in parts of the world, people sleep with their chickens, right? And that...
Well, yeah, but the majority of the world doesn't do that anymore. Whereas before,
even in the developed world, we do live with some wild animals.
Yeah, but she's pretty much domesticated.
Tell her that.
Domestic cat versus. My friend had a feral cat.
Yeah, I think this one.
And that's totally
close.
I was going to say that's like he was not when his husband brought it home. He was not prepared.
Other than using the
totally
litter box,
different.
this one is pretty wild.
So primates, primates have had bornaviruses for tens of millions of years.
Yes.
So that's basically the study. 131 species, vertebrates. These are vertebrates that have endogenous Borna elements in them. They can go back almost 100 million years, 96 point something million, but can round it off. Sounds really good. And they say, these virus-host relationships probably, these are over geological time scales, right?
Yeah.
It's a long time, and a lot of things can have happened. So we don't know the whole story. And this is a great point they make. These endogenous elements only provide limited snapshots, right? Because first of all, they're really rare when they happen. It's not like this happens with every infection. And so a lot of the viruses probably never endogenized. And so they may have disappeared. Their host may have disappeared. And so this is basically an incomplete fossil record. Well, I guess every fossil record
is incomplete. But yeah. I actually have a little quibble with something you wrote on your blog about this paper. You start off by saying all human viruses arose from spillovers from other animals. And as soon as I read that, I said, well, what about the ones that were in us when we evolved as a species?
Well, they came from someone else, right?
Right.
So,
but
and eventually...
And then we became humans, but we brought some viruses along with us, I'm sure.
Yeah, but they're all in some distant past. They were all a spillover at some point. In other words,
right?
No new viruses arose in us. In fact, probably no new viruses have arisen for millions and millions of years, right?
Okay, it depends on how you define new viruses.
Yeah, that's all I meant by that. I know that when I wrote it, I said someone's going to quibble with this.
Yes, here I am.
But Homo sapiens got viruses from other Homo species, as well as, Australopithecus passed it down.
And at
to
whatever
them as well.
arbitrary moment we say we became Homo sapiens, we already had some viruses with us.
Exactly. We had some but they came from animals.
They came from,
because animals, I don't know, it depends. Homo, only starts at a certain time. So the virus is around long before that. That's what I meant.
But this gets to the problem that species are arbitrary, and so we can't really define a starting point for our own species either.
And they also say...
Yeah, but I'm sure, you have it. I'm sure, if you looked at, fish or something, which is far older than we are, and you go back all the way to the cartilaginous fish and stuff. I'm sure that there are endogenous elements that you can,
Oh, yeah, sure.
Yeah, I imagine the reason they picked, obviously, vertebrates and bornaviruses, I think, is that this is what the database has a lot of.
Yeah.
Well, but I'm sure some of our stuff came as ours.
Yes.
If you go back and you try and compare it to the fish,
some of our stuff came from when we were fish.
Yeah, I was going to say you can probably trace all the way back to when we came out of the water, when we were not vertical, when we became vertical, when we stopped walking on four limbs, blah, blah, blah.
Going back to the episode we did at Penn State, what was the guy's name? Works on coral herpes viruses?
Forrest Rower.
Forrest Rower, which was a fascinating, I mean, his work was just really cool. And he was talking about herpes viruses that infect corals. And their biology is strikingly similar to the herpes viruses that infect humans. It basically makes a cold sore on the coral. So we've had herpes viruses with us, since before we had backbones.
Yeah.
So there are
kind of like my
there are
point,
bornas of fish and snakes, obviously, but there are probably not enough genomes to do this kind of analysis.
People haven't sequenced enough fish.
Yeah.
I mean, what's kind of interesting to me is the fact that, if you say that there's parts of all these proteins, that we're still deficient in an RNA-dependent RNA polymerase. We have a DNA-dependent RNA polymerase. We have a DNA-dependent RNA polymerase. We have an RNA-dependent
DNA polymerase.
Polymerase, but we don't have an RNA-dependent RNA polymerase.
Maybe it causes too much chaos.
I don't know, but kind of is interesting in that.
Right, but our we've got so many systems built around double-stranded RNA as a threat that
that maybe
every time we've picked up an RNA-dependent RNA polymerase, it gets eliminated.
but it may also be why that we have, whatever. I mean,
Interesting.
you have double-stranded RNA structures that are not
True.
triggers.
Right.
So we have also the other aspect I wanted to summarize is that these lineages, and they can track the movement. We didn't really talk about this, but they can track the movement of lineages among the continents. And so they're thinking that, infected animals moved and brought the bornaviruses with them, and they could infect other species as well. And this includes bats, by the way. Bats get infected. They even Rhinolophus bats have these endogenous elements. And Rhinolophus we will encounter in the next paper.
Yes.
But they say, so what's one thing that's interesting is that the N protein, they find the most integrations of that, far more than all the other mRNAs. That's quite interesting. In fact, I think we've talked about some, may not be Bornas, but other viruses where they believe the nucleoprotein is actually produced and it defends against infection in cells. We did that paper a long time ago.
Right.
So that's kind of interesting. And they say the study also implies a large number of EVEs derived from unknown ancient viruses are hidden. So they found a lot of sequences, but they can't tell what they are.
Right.
Maybe they're viruses that went away, that became extinct.
Right.
So
or
the
sequence has just evolved too far away from what it originally was.
So they say we should extract nucleic acids from more extinct animal specimens to try and museums have lots of these, so that would be a good source, right?
It's tricky, but it can be done. A lot of the museum specimens are formaldehyde preserved, which is not great for, if people had known that we were going to need the nucleic acids, they probably wouldn't have used that. But it is actually, there are people who've figured out how to extract usable sequence. You're not probably going to get whole genomes.
No, you get very small pieces. So I think Paul Planet, when he was here, used to do that. Mm-hmm. But my technician's brother used to do that for the Museum of Natural History on birds.
Right.
So when Audrey was here, Jack did something like that.
The retrovirus that's infecting koalas in Australia, they went into museum pelts, koala museum pelts, and they found sequences 150 years ago, so they could get an idea of when it emerged, because that's thought to have come over from rodents. So sometimes it works. It's a whole field of ancient DNA. DNA is easier than RNA, of course, but this is what you're looking for, the DNA, and then you look for the endogenous elements. Forget about RNA, right? It's not going to last very long.
Well, people did. It lasts long enough to get the 1918 flu.
Yeah, I think that's the oldest one, right? That's the oldest RNA recovered.
I thought that there was something slightly older, but it's...
there might be an older...
In a long time.
There may be a 1,000-year-old RNA virus and some poop from an animal, I seem to remember.
Yeah, but I haven't thought about it in a long time. I work on a more contemporary virus. What can I say?
Oldest
RNA
virus recovered. Let's see if we get a hit. Oh, look at this. The oldest is 750 years. This, a 1,000-year-old RNA virus. They call it 1,000-year-old. That's by Marilyn Rusink. They got it from a plant, corn. Zea maize chrysovirus 1. Yeah, 1,000-year-old. In 1,000 years of maize cultivation, this virus has undergone 3% divergence. Okay, 1,000 years.
Right.
I guess there are a lot of maize samples. A lot of maize samples are preserved, right? Cobs. They extracted RNA from cobs. Well, here you go. This is a double-stranded RNA virus. That explains it, right? A little more stable. Less stable. Okay. Borna.
Well, you didn't specify that you were only looking for single-stranded.
No. RNA is RNA, isn't it? Like DNA is DNA.
I don't know. I thought we were recently told that wasn't true.
Well, I made that argument a long time ago. Anyway, let's move on to our second paper. This is now moving to SARS coronaviruses. This is a journal preproof from Cell, and it is Identification of Novel Bat Coronaviruses Sheds Light on the Evolutionary Origins of SARS-CoV-2 and Related Viruses. It's from a number of institutions in China and the University of Sydney, which is the site of Eddie Holmes, who's on the paper. You have universities of Shandong, Chinese Academy of Science, et cetera, Chinese Academy of Science, Beijing, and Hui University. I got a long letter the other day of how I should pronounce Zhongli Shi's name, because I apparently massacred it. I do not doubt that I massacred it, as many people massacred my name.
The authors on this, first author is Zhao.
But not Dove, right?
Senior author is Shi, and Vincent mentioned a couple of others. Do we have any co-first or co...
Let's see. Number 9, these authors contributed equally. So 1, 2, 3, 4 first authors. Zhao, Ji, Chen, and B. And then I think we have a lead contact is number 10, Weifeng Shi. And Eddie Holmes is just before that. I suspect Eddie Holmes did a lot of the computational analysis here. Okay, so what this is some field work. Now, people have always asked, are we doing field work and looking for viruses in bats in Asia? And this was done between May 2019 and November 2020. So, before and coincident with the pandemic. So, as you know, bats are important sources of viruses. They're 20% of all mammalian species. And I didn't know that rodents are 40% of all mammalian species. Wow, because they said they're the second most diverse mammalian order after Rodentia. So I looked up, and rodents are 40%. Yep. A lot of rodents out there.
Rats, I didn't know that.
And a lot of
mice, for sure. So as you know, many coronaviruses have originated in bats, not just SARS-CoV-2, but others. So bats are being examined, have been for a long time.
Rodents have also been sources of many other viruses, but
yeah, and I think 2 of the common cold coronas came from rodents.
Probably rodents.
One from a bat and one from a cow. Anyway, so in this paper,
the virus that's in Colorado,
Hantavirus.
The one
was that Colorado?
That was the one you were thinking of. There was a hantavirus infection in Michigan recently. The first hanta in Michigan. Yeah.
Oh.
These come from rodents, Paramyscus maniculatus, the white-footed deer mouse.
Yeah.
Anyway, so here they're looking at coronaviruses in bats. So let's summarize what we know. We have SARS-CoV-2. What do we know in bats that's close to it? So we have RATG13, which has the greatest overall sequence identity across the whole viral genome. The whole genome is going to be one better than it, but not over the whole genome here today. And that was identified that was sampled in 2013 in Yunnan. And so that's RATG13, very famous. And then and that's from Rhinolophus affinis, okay? RA, and that's where the RA comes from. And then we have RMYNO2 from Rhinolophus malayanus, closest relative in ORF 1AB, which is the very long ORF at the first half of the genome. And it has this kind of a similar insertion at the S1, S2 cleavage site. So SARS-CoV-2 has a furin site. And this is not a furin site, but it's getting there, right? So it's a similar insertion. So that's also quite close overall, but not as close as RATG13. Then we have the pangolin viruses, the Malayan pangolins, from which SARS-CoV-2-like viruses were identified. 2 lineages, one from Guangdong and one from Guangxi. And interestingly, the Guangdong, they have lower overall identity to SARS-CoV-2, but in the receptor binding domain, the Guangdong pangolin isolates have 6 amino acids, the 6 critical amino acids for binding ACE2, the same as SARS-CoV-2. So that's quite interesting. We have a distantly related SARS-CoV-2 from a bat in Japan, Rhinolophus cornutus, and then 2 novel beta coronaviruses from bats in Cambodia. These were sampled in 2010. They're 92.6% identical with SARS-CoV-2. 5 of the 6 receptor binding domain amino acids are identical to SARS-CoV-2. And then we have the coronavirus from Thailand, which we talked about on TWiV, from the irrigation pipe.
Right. And
this is from Rhinolophus acuminatus. That's found in June 2020, and that is very similar to RMYNO2. So you can see from Japan all the way down to Thailand, you have bats harboring SARS-CoV-2-like viruses.
So that would be the 4,300 kilometer distance that Linfa has been talking about.
And so this viruses could have originated in any of these 4,300 places.
So let me get this straight. So, and then within this population, but not in the area of SARS-CoV-2, there's a virus that has 6 amino acids that look exactly like SARS-CoV-2 in the RBD.
Right.
Okay.
Yeah, this is really.
So I really see all this other stuff as just came to us like by divine intervention, and we created a virus called SARS-CoV-2. Okay, that makes a lot of sense. So this is
really reminiscent of the story of tracking down the origins of SARS. SARS-1.
Well, yeah, that's why I picked this
and
paper.
this is that's exactly what happened. We got closer and closer versions of, well, this piece is here, and finally we get down to one cave where all the pieces of SARS, and I don't think we actually ever quite got to the point of the exact bat and the exact virus.
No.
But it's all the pieces of it are in one cave with the bats hanging out together in that cave, and we know these viruses recombine like crazy, and so all the ingredients are in that one spot, and that's where we're sure it came from.
Right, but here, that's one scenario, right? But the other scenario is bat. You take bats bite something else or whatever, and the animal that is is then the intermediate host is some kind of honored sacrilege thing that you're going to pay $1 gazillion on. So you ship it to some market 4,000 miles away, and you sell it something.
There's a civet.
You sell it to somebody else. Blah blah blah. They breed whatever. I mean, you can imagine hundreds of different kinds of combinations of how.
Well, and we know that in the SARS case, we had an intermediate host in the civet. So it wasn't the bat cave gets you the ancestor virus, and then the civet gets you the rest of the way to the spillover event.
Yeah, but I'm just saying that, but I'm just saying that you could, move the animal thousands of miles
easily,
right? So,
no, I don't think the location. I'm not going to reopen the location naming thing, but I don't think the location of a particular bat is necessarily informative in terms of the origins of the pandemic, because people move these things all around, and of course the bats, move themselves.
Well, that's my point.
I would argue that if you're interested in ecology of viruses and their hosts, then the origin matters.
Does matter,
right?
Yes, absolutely.
So that kind of information.
Right, but I'm not arguing.
No, I'm just saying. I want to just point it out that I don't need viruses named to incriminate any country, but if you want to know ecology, you need to have that information, right? And today,
but it doesn't need to be.
That's fine, but somewhere it has to be stored so that you know it's associated.
Sure. Well, you catalog it, right? You catalog.
So, but that's the SARS-1. It's not irrelevant. It's relevant for people who are interested in ecology.
Yes.
For SARS-1, the civet isolate was 99.9% identical to the human isolates. Okay,
right.
But as for the bat ancestor, right, the cave had all the genetic pieces of that, but no one had one virus that was the ancestor.
Right, but you can imagine that if the intermediate is two different kinds of animals, right? You can imagine that you take viruses from one bat cave in, I don't know, Mars, move them to some market on Jupiter, have another seller come from, Mercury with his little animal critters that live in the same cage as the critters from Jupiter, and then the viruses reassort, and then you go to Saturn, and now you have
absolutely. It could be
something else, right? So you don't
next thing you know,
necessarily
you're
need
up to
to always
Uranus
be in
and
the same
viruses.
cave. Exactly.
We don't have to have.
But you don't need to be in the same cave. Is
Totally.
really my
You don't have
point.
to have the same scenario as SARS-1. Absolutely.
No.
Nature is varied and diverse for sure.
I'm just there are parallels here in the way that we're triangulating on it.
Right.
Yeah.
Right. But I'm just saying that in reality, I can come up with six different ways of that.
Yeah.
I mean, I haven't even factored in the fact that like some viruses pick up cleavage sites from host genes, like influenza, right? Because we don't really understand fully how recombination, reassortment, and all of those things work. And so if we don't understand that for flu, which we've been studying for a gazillion years, how are you going to say that we understand it perfectly for something that we've been studying for 5 minutes?
But the point of this study is that you slowly sample more and more. I mean, this is a very small area. This is a 1,400 square hectare part of Yunnan province called Mongla County. But you do more and more of this, and you get closer and closer. By the way,
I thought that they also looked at Laos.
No, that's.
I thought that there was something about
the viruses
them crossing the border.
No, the viruses.
Into Laos.
Viruses are only in Yunnan province, but the bats have a very broad range.
Right. So they do ecological modeling on the bats.
Yeah,
and that's a projection of how far these bats are spread. But the virus samples they're collecting are just in this. Works out to about 3,000 acres. So imagine the size of a large national park or a small to medium sized national park.
They call it a tropical botanical garden.
Right.
So this is very interesting. So Mongla County, Yunnan province. Mongla is the name of another virus that we have talked about before. And Kathy knows because she pronounces it the right way and I'm not pronouncing it right. So the sampling was done between May 2019, November 2020, 283 fecal samples, 109 oral swabs, 19 urine samples. And in the graphical abstract, they have picture of poop, oral swabs, and urine. It's very funny, very cute.
Yes.
And mostly horseshoe bats, but some others. And then they did RNA sequencing, essentially. They initially pooled them, did RNA sequencing, and they eventually find 9 beta coronaviruses and 17 alpha coronaviruses. And they note that one of the pools of fecal samples was previously used to identify RMY NO1 and NO2 that I mentioned before. So they name these in the same way, where the first 2 letters of the bat species, YN is Yunnan, and the final number is a serial number. So, okay, let me ask you, with no bias here, is YN okay, or is
I think YN is okay.
Yeah?
It's okay with me. Because the only people who are going to parse that out are the people who are interested enough in the virus that they're going to understand what it means.
Now, so it might be a good solution just to have 2 letters, right?
Yeah.
But, of course, if it were NY, then you'd know.
No, not necessarily. A lot of places could be NY.
Yeah. Okay, so 4 of the 7 beta coronas were related to SARS-CoV-2. The other 3, SARS-CoV-1.
And I want to mention the numbers here just again for context, because they're sampling 342 bats total, and they pull out 24 coronavirus genomes. I mean, it's a lot of work to sample that many bats, but in the grand scope of things, that's not that many bats.
No, and you get a lot of viruses.
You've got a couple dozen novel coronaviruses, new to science, pulled out of these bats, and several of them are very closely related to SARS-CoV-2. These things they're everywhere.
Yeah. And they actually go back, after they have the genome sequences, they design PCR primers, and they say, can we find these in individual bats? Because originally they pooled the samples, right? And they can. They can find, not huge numbers, but, 1, 2, 3, 6, 8 bats that are positive for any number of these novel viruses. So I think it tells you that you go out and sample, you're going to get results, right?
Yeah.
Because the bats have a lot of viruses in them.
Oh, you want a sample?
I do.
Yes.
I would go if it were up to, look, if someone said we need someone, I'd go.
I'd have to get my rabies shots first, but sure. Yeah.
If you really need a worker to go, I'd do it because I think it's important. But they have plenty of people to do it there. I don't have to do it.
Yes.
I don't have to do it. I think it's really important to do it.
This is
and not just in Yunnan,
work.
not just in Mongla in Yunnan. You have to do it in all the surrounding countries, right, as well.
And not just going after the origins of SARS-CoV-2. I mean, this is something that we really ought to be surveying globally everywhere.
Yeah.
In many, many different species and just...
Well, that's why we have mice from New York City and surrounding areas sitting in our freezer waiting for me to do something with them.
So let me tell you a little bit about these viruses. RPY, so remember, RP is the bat. RPYN06, 94.48 sequence identity to SARS-CoV-2, which makes it the second closest relative to date.
And that's overall sequence.
That's overall. We're going to see. Now, as you'll see, there are parts of these genomes that are really close and then parts that are different, which is because these viruses undergo recombination, right? So the similarity varies across the genome, and you can use that as a way of figuring out the recombination pattern. For example, RMYN02 has 97% identity with SARS-CoV-2 in the 1AB open reading frame. And RPYN06 has 97.19% in the 1AB, so more than the overall genome similarity. Interestingly, RPYN06 has the highest identity with SARS-CoV-2 in the RNA polymerase gene, the ORF7A, ORF8, the N protein, and ORF10, 100% identity in ORF10. But only 76% identity in the spike gene and 60% in the receptor binding domain. So a little bit of recombination going on there. And then they have some others, which, the other interesting part is that RSY04, RMYN05, and RMYN08 were 99.96 identical to each other at the whole genome scale. So these are all very similar to each other. Say viruses of the same species. And interestingly, they were collected from different bat species, right? RS and RM, different bats. So pretty cool that these viruses can infect different bats.
Yeah, and as they point out elsewhere in the paper, it's very common for these multiple bat species, especially in the Rhinolophus genus, to be in the same cave together or very similar locations together. And they're, as we've said many times before, these are social animals, and it's not surprising that they're able to spread the viruses. It is interesting that the virus is able to just cross species like that so easily.
So they do, of course, phylogenetic analysis. They do whole genome, and they do individual genes. Again, SARS-CoV-2 most closely related to RATG13. RMYN02 and the Thailand strains are a slightly divergent clade with RPYN06 at the basal position in the clade. RSYN04, 05, and N08 group together, and they cluster with the pangolin viruses from Guangxi.
Yeah.
It's interesting. And then the others, N03, 7, and 9, fall within the SARS-CoV-1 lineage, which is a separate branch on that tree. That's the overall genome. But if you look at individual genes, you get different patterns. Different pattern with the RNA polymerase. I won't give you the details, but it just goes to show that genes move around from virus to virus, and they have a different evolutionary history.
And this paper is open access, so if people want to drill into that, they can go ahead and download it.
They do phylogeny based on RNA polymerase, the spike, and you get different results.
Well, they also do other accessory proteins
Yep.
like
Yeah.
ORF8, the loss of ORF10.
And it's
some
really
cases.
important to do those sorts of gene-by-gene phylogenies because, as we've discussed, there's all this recombination going on. So really, we are interested in the evolution of individual genes and how they came down.
So then they're interested in the spike and its interaction with ACE2. So they do a number of experiments. They do some structural modeling to see how similar these are. But what I want to tell you about is they actually made the RBD, the receptor binding domain, from these viruses. So they made the protein and then looked at its ability to bind ACE2.
Right.
Of course, SARS-CoV-2 RBD binds very nice with KD dissociation constant 11 nanomolar. No binding with the RBD of RPYN06, but RSYN04 did associate, although the KD was in the micromolar range, so not as strong as SARS-CoV-2. Let's see. Then ecological modeling of the bat species. The Rhinolophus species throughout Asia. And they look at different factors that control the abundance, like temperature, seasonality, continentality.
Continentality, I like that. How close they are to the coast. It is. Yeah,
important.
it turns out that's like possibly the most important factor on average for where these species locate.
Yeah.
Rainfall, evapotranspiration seasonality,
Yep.
and
Hey, you don't want
Yeah.
a
You
drought.
have wet season,
And
dry
You don't want to
season.
be dying of thirst.
I like Emberger's
I don't want-
pluviothermic quotient, a measure of climate type. Distance to bedrock, an indicator of caves. That's cool. And local climate that influences diet availability. So they couldn't model Indonesia because they didn't have enough data, but most of mainland Southeast Asia's tropical forests have a high diversity of rhinolophid bats, 23 different species, with the largest hotspots from Laos, South Laos, and Vietnam to southern China. I bet that's where you'd find a lot of very highly related SARS-CoV-2 viruses.
Yeah, I wonder when we discuss this constantly, who says you have to go south?
Is that what you said?
Yeah.
Yeah,
Yeah.
especially after you had Peter and others on. I really I think it's great that they went to Wuhan, but I think you'll find it'll be a richer treasure trove
Yeah,
to
I
answering the
agree.
question if you cross the southern
Well, and
border.
this paper,
Go
they're
into
looking.
Thailand
They're looking in Yunnan, in southern corner of China, and what I'm getting from this paper is a big signal that's saying you're getting warmer. You're getting warmer.
Yeah.
You're getting
Yeah,
closer.
that's-
and
yeah.
you're exactly right. You've
But-
got to go. You've got to go into Laos and Vietnam and adjacent countries, and not necessarily go there, but collaborate with people there and sample bats in those places.
Yeah, I mean, I just, for whatever reason, when for months that Vincent and I have been talking about this, I've been I highly doubt that we're in the right spot. Like, you have to go south.
Yeah.
And it might have been started when I saw Lympha's paper.
They also say that
And he talks about the 4,300.
Yeah.
they also say that several rhinolophids extend northwards.
Yeah, there's a whole pocket
Yeah.
of them in southern Russia,
Yeah.
and I referred to it as lower Russia or something. And somebody said, something, whatever I said the other time I talked about this. But there are actually Rhinolophus bats also in Hungary and Transylvania and stuff. But there's no Rhinolophus bats, if I remember correctly, from what Simon said when I was talking to him, in South America, I think in Africa, and definitely here, there's no Rhinolophus bats. We don't have the right climate
Right.
and stuff. And so I said to him, so basically bats are like mosquitoes.
Yes.
So there are other rhinolophid species as well, and they say one of these, Rhinolophus theno, has both SARS-CoV-2 and SARS-CoV-like coronaviruses. So that's the story. I think 40 of the 100 libraries gave them coronaviruses related to SARS-CoV-2 and SARS-CoV, and they say these viruses closely related are still circulating and maybe at high frequency in some regions, right?
Yeah, and we've talked about how many bats are
circulating?
migratory, but they point out in this paper, Rhinolophus are forest bats that probably don't make really, really long migrations. They may seasonally move someplace for mating or for food, but it's going to be over relatively shorter spaces. So I think to Amy's point, the idea would be that this SARS-CoV-2 came from some combination of local bat dynamics someplace in Southeast Asia, possibly south of China, and maybe crossed the border in a bat, maybe crossed the border by humans, and then humans transporting stuff, might have been a bat, might have been a civet, might have been a pangolin, ended up getting to SARS-CoV-2.
Yeah, but just to embellish that a little bit more, Alan, that's pretty close. But if you looked and learned the lessons that when we did the NEPA paper, because those bats only go like 20 miles, right? And how that NEPA has moved out of the NEPA belt into whatever region of India that Dixon went to, Kerala or whatever it was. I forgot. But I think more along those lines of how it's been moved from the different Rhinolophus, either hives or nests or whatever, or species, is more...
By getting from a bat into something that people move around more like a pig.
Yeah, but I didn't think that the NEPA paper said that it moved via a pig. I think it talked about how the bats socialized, and that's what trafficked the NEPA out of the NEPA belt. And so that's what I think it's more along those lines that we don't understand. Well, yes, they might go 20 miles, but we don't really understand the social
interactions. They might meet up with another species of bat in that same area 20 miles away because that's where the food is, and then that species migrates north, or it's like a relay race.
We don't really or a game of telephone.
So one of the other conclusions they make is that it looks like there's virus transmission between pangolins and bats, so that they've got these closely related viruses from pangolins and bats, and that could explain why the pangolins are infected, because we really didn't know before. They say this is remarkable phylogenetic and genomic diversity of bat coronas in an area to which humans may be routinely exposed.
Yes.
And further surveillance efforts are essential and should cover a broader range of animals in this region.
Yeah.
Not just bats and pangolins. I think that's a good idea. Yeah, for sure.
Really? I was thinking we should have more political commentary.
Yes, right. We need more pundits.
I was thinking, why do we need to survey more when we have more? We could have more political.
We could just have it as a matter of opinion.
And
that would be so much more.
Well, it's not. Well,
yeah, we could have politicians talking about coronaviruses. Right. That makes a lot of sense. Right. Actually, we have an email about that. That's what Amy is referring to. All right. I thought that was a very nice paper.
You can even go, I mean, it doesn't necessarily have to have an intermediate host either to take the counter argument. You can go from bat to human, and then the human travels because they do that.
Yeah.
Right, but even clearly you don't need to go any intermediate host because I forget which one. I know it ends in 406. Already binds 404. Yeah, already binds ACE. You tweak it a bit. I mean, maybe the binding, you only looked at the RBD. But maybe in the context of that whole protein, the binding goes from micromolar now to nanomolar.
Sure.
You really don't know what the context of
we need more sampling.
The full-length protein.
Don't you think we should have been sampling more after SARS-1?
Yes.
Kind of.
Oh. You want to go back and say, you want to look back and say with 2020
Well,
hindsight?
no, even before SARS-2, I think a lot of people, including maybe even me on a couple of occasions, and certainly Vincent, said that we ought to be sampling more. And
Well, that's why we have, as I said, that's why we have mice
Yes.
from New York City and the surrounding areas in our freezer, courtesy of Ian. And if I had, another pair of hands and 4 more hours every day and another day to the week, I could actually answer some questions about what is sitting in the freezer instead of just accruing it. Plus, I have people samples from Ecuador.
I think the point is that rodents in the U.S. are very undersampled. We have very
Sure.
little understanding of what viruses are in them, and needs to be done.
We have very little understanding of what viruses are in
Yep.
any
Yeah.
animal. in
But
the
we
US.
focus on the numerous rodents and bats, right? But I also think birds are important because obviously for influenza viruses, they may harbor other viruses and they're mobile.
West Nile.
Yeah. So, okay. Cool story. Thank you for suggesting that paper, Dr. Rosenfeld.
Yeah,
Let's
it is
let's
a
do
cool
some, and
story.
there'll be more, hopefully. Let's do some email.
I'm sure that they haven't.
First one is from David Quammen.
I've heard of him.
I just finished listening a little belatedly because of drinking from the fire hose. When I was reading that, I think because of drinking.
Because of drinking. Oh, okay. Well,
To your May 30th TWiV with Bob Gary. I spoke with him several months ago too, but your conversation was extremely valuable to me and others on many points and so illuminating to our current storm of misleading articles and fuss. Thank you much. A great service. And I agree about Amy Maxman's piece. Onward. High appreciation, David Q. Amy Maxman, of course, with the Nature article saying, hey, can we try and be civilized about this and not alienate everyone?
yeah.
She's had 2. She's had 2 articles about the origin.
Amy, can you take the next one?
Sure, I'm all right. Dear Twibbers, I'm writing you from Grey Paris, where it's currently 28°C and raining. You discussed in one of the latest episodes that it is not correct to associate mutations with a new variant of SARS-CoV-2 to better transmission, and that for flu example, we talk about fitness of viruses, never transmission. I don't quite understand clearly the difference. Isn't better transmission mean better fitness? No. Although does better fitness mean better transmission? No. What does fitness exactly mean? The mutation causes protein of the virus to be more stable? Could be. Does it mean that the virus becomes more adapt to the host, replicates more, maybe escapes some immune responses? Could be. If the mutation causes the virus to replicate more and for a longer period, does this mean it would possibly increase its transmission, higher virus titers from an increased transmission time window? Thank you for your clarification. Best Amal. So can she just read Vincent Munster's paper where they look at one of the variants and they show it has better replication and it just sheds better, but it doesn't necessarily imply anything about transmission?
Yeah, I mean, it could. So Amal says if it causes more replication for a longer period, could it possibly increase transmission? Sure. And
Yeah, but you have to
prove it. And so the paper Amy is referring to is a preprint from Rocky Mountain Lab where they show in a non-human primate there's slightly more shedding with the alpha variant. Okay, so does that prove that it's more transmissible in humans? No. So you could do a transmission study in an animal, but that wouldn't prove it. So, you need multiple lines of evidence and you also need human data, I think. But you're thinking correctly, Amal.
Yeah.
You're
I think
thinking
the simplest way to put this is increased transmission is one way that a virus could achieve increased fitness.
Right.
Okay. It would be a fitter virus. It would be better able to spread if it had increased transmission. However, there are many other ways that a virus could increase its transmission, including not increase its transmission, increase its fitness, including a longer asymptomatic period. So it's not more transmissible. It's not more able to get between hosts. It's just active in the host for a longer period of time, and the host is asymptomatic and therefore more likely to spread it. That would increase fitness.
Are you going to do Nevin's paper?
I think we should at some point, yeah.
The ability to evade an immune response would increase fitness. There are many, many other ways
Right.
that a virus
So
could increase its fitness.
Right. So his conclusion basically is that the spike changes had no effect, but it's this change in ORF9A, which is which makes it a very potent antagonist to the innate immune response.
So the issue is that virologists use transmission in a very specific sense. We don't just mean that the virus becomes more prevalent in the population. We mean that it's happening with any of the variants that people are referring to as highly transmissible. Yes.
Right.
And I wish that people would adopt them more in wider discussion of these, because when you refer to something as being highly transmissible, it carries the implication that the non-pharmaceutical interventions that people were taking would become less effective, which is not supported by the data. Right. I'm explaining to Amal.
Yes.
Well, yes, I understand that.
Yes.
Oh, I was going to say, I understand.
It's hard to get that usage into.
It's very hard to. I don't know. It's hard to. You know, there's a colloquial definition.
And the problem with fitness is because we don't teach evolution properly, people misinterpret fitness to think that the virus has big biceps. And so we can't. No, right.
Wow.
That's a separate.
You're not allowed to.
It's a related but separate problem.
You're not allowed to teach evolution. Right?
I could, yes. I can take the next one. Jim writes, I've been a loyal listener throughout the entire pandemic. How do you respond to the discussion of lab leak theory on the Joe Rohan podcast with Crystal Ball and Sagar and Jetty? Is your hard stance against the lab leak theory due to inherent bias? Is it possible there exists to modify a virus that quickly? I really respect the objectivity of Crystal and Sagar, and they seem to have at least evidence of a conspiracy to shut down any discussion of the possibility of a lab leak. I would love to hear your response, as it will help drive my thinking of what really happening. Okay, Jim, so I want to take a couple of things here. First of all, I do not have a hard stance against any theory, including the idea that it came from a lab. I do, however, insist on evidence before people go out committing libelous presentations of arguments that slander somebody who I respect, or really that slander anybody. So this idea of the lab leak is making a specific and very serious charge against identifiable individuals, as I've said repeatedly. And I have a problem with that when there's no evidence, which there isn't any evidence that that happened. There are quite a number of reasons to believe that that didn't happen, not least the behavior of the scientists who would allegedly have been involved in this, who, you know, opened their facility to a team of foreign visitors, experts who were allowed to wander around talking openly with folks in the facility, and that's just not what you do if you have secrets to keep. So there's all that. I don't know this podcast. I don't know Joe Rohan. Somebody is Joe Rogan. Okay. I've at least heard that name.
I think it's Joe Rogan. It should be Rogan,
has helpfully
right?
added the credentials for Crystal Ball and Sagar and Jetty. Thank you very much. Both pundits. I don't see any particular virological.
I did.
Or epidemiological qualifications. These are folks who are professional opinion havers, which I guess is fine. Nice work if you can get it. But I don't. Right. So one is on the left and one is on the right.
Well, they're liberal. They were liberal.
That doesn't constitute objectivity. It just constitutes opinions from different
that's
political
why their
spectra.
podcast is balanced.
And I don't know what evidence they claimed to have of a conspiracy to shut down discussion. I think there's actually been quite a lot of discussion of this, so that conspiracy has really failed.
We're not trying to shut down anything. We're trying, as Alan said, there's no evidence. No. We don't have an inherent bias. If there's no evidence, we're scientists. We look for the data. There's no data, and there are plenty of data that it came from nature. For example.
Right. I mean, we've got tremendous precedent for this coming from nature. We don't have any evidence that it came from the lab. So before people go making these slanderous accusations that it came from the lab, I think they need to come up with some kind of evidence. And evidence is not, oh, it's got a furin cleavage site that somebody could have maybe kind of possibly put in there if they'd done this, this, and this.
Right.
That's not evidence. That's wild-ass speculation.
But as I said, like flu acquires a furin cleavage site that wasn't there naturally. That's what makes it
and so we have precedent for all this happening
RSV,
in nature. And we don't have any reason to believe that this would have been done in a lab. And in fact, there are a lot of reasons to believe that nobody would have thought to do any of these experiments because they wouldn't have made any kind of sense. So yeah, that's our bias, if you will.
You have to put too many pieces.
You have to put too many pieces together. And I have a human behavior objection to this notion because at the very least dozens, and possibly at this point hundreds of people would have to know if it had come from the lab, right? Somebody had to do those experiments that allegedly would have led to this. And there are too many people who would have to have been involved and who would then have to not only agree to keep this secret, but keep their stories 100% consistent, even in the presence of expert questioning. So when Marianne Koopmans et al. Went there and were visiting and were saying, we're asking them questions, did you work on this? What did you, somebody would have screwed up. There's no way you could keep all of that straight and not have a slip up.
I think Jim is also, is it possible to modify a virus that quickly? Now you're conflating two ideas. So one idea is that it was made on purpose, which is absolutely not true. That's preposterous. And if Crystal Ball and Sagar and Jetty suggested that, it's because they have no idea how to modify and produce viruses.
Well, let me tell you, I do, and we fail at it 99.9%.
And when you succeed, you don't do it this way. No.
I was going to say, but let me tell you, my technician in undergrad are trying to do it.
So Jim, that was not made in a lab, and Crystal Ball and Sagar are not the people to talk about it. I'm happy to talk. Any of us on TWiV are happy to talk about it. The other accusation, and that's really what it is that it escaped accidentally from the lab, for which there is no evidence. And as Alan said, sure, it's possible, but there's no evidence for it. And Zhongli Shi has said she wasn't working on any material that had this virus in it. And I believe it. Now, if you choose not to, that's not science. She said she didn't have it. There was no one before December 2019 with SARS-CoV-2 anywhere near the lab. And I think that makes perfect sense. We're now seeing it's throughout bats in nature. So,
well, we're also seeing that the virus actually we're starting to see that cases were actually before.
Yes, but no one had the virus in the lab at the time. That's what.
Well, I understand that.
Right. I'm sure we will see that it was circulating in October.
Cases were earlier.
Yes, for sure.
Right. So to say that they made a virus in December and cases were circulating in October, I think you might want to revisit when the calendar months occur.
I mean, making the viruses, what would you use to make it? RATG13? Too distant. Not the ancestor. And nobody had anything else. So, Jim, I think you need to listen to virologists. We are not biased. We just go by the data. And you may see that as bias, but it's not.
And conspiracies are not unheard of, but successful conspiracies of this scale are, I believe, impossible. Carol
writes, what a wonderful podcast. Is it my turn, right? Did you read that last one?
Yeah.
What a wonderful podcast you produce. I began listening during the early part of the pandemic, was instantly hooked. Thanks for entertaining and educating me all these months. Your picks generally delight me. I'm a bit of a software nerd, so I was pleased by Vincent's choice in 768 of Steve Gibson's rant. The rant reminded me of the late, great Daniel Kaminsky's comment about internet security as related in his New York Times obituary. Quote, The internet was never designed to be secure, Mr. Kaminsky recalled in a 2016 interview. The internet was designed to move pictures of cats. We are very good at moving pictures of cats, but he added, we didn't think you'd be moving trillions of dollars onto this. What are we going to do? And here's the answer. Some of us got to go out and fix it. Pictures of cats. The 2016 interview is on YouTube. Just search for Hacker History, the Great DNS Vulnerability of 2008 by Dan Kaminsky. Note that I am not sending you a link since you don't know me. And why would you open a random link in an email? Very good. It's a great point. Anyway, the interview is done as a cartoon and is one of two produced by Duo Labs, part of Duo Security, now owned by Cisco. Thanks for telling me all about viruses. I mean, that was the point of the Gibson rant, that you can click on a link in an email and all kinds of things can happen to your computer. So don't do it. And I do not, my email comes in as plain text, no HTML. And if there are pictures, they're not downloaded. So if there's a link, I can copy the link and look at it before I would click on it. That's what you should do. You shouldn't have pictures automatically downloaded because that's how they can put trackers and things in.
No.
Thank you, Carol, for that. Amy.
Emma writes, hi, TWiV. Thank you for all the great reporting you've done. I especially appreciate your skepticism regarding the much hyped new variants. This brings me to a New York Times article about the disappointing CureVac results, which suggests that the cause for the low efficiency, 47%, is likely due to the variants. But it's not the same technology. They used unmodified mRNA, which has a harder time getting into cells, and they used a very different sequence. I'd be really interested to hear your interpretation of this result, since the temptation to blame everything on super variants seems to preclude asking lots of interesting questions. Best regards, Emma from Palo Alto, where it's 93 degrees. And she sends, one, the article of record from the New York Times about CureVac, and two, a random blog post my boyfriend sent me with much more information about the differences between the vaccines.
Yeah, the CureVac vaccine is a totally different product from the other mRNA vaccines, and I don't think the variants are the problem here.
No, they're not. It's not the problem. So not only are they not the problem, but I believe that Theodora Paul, was David Ho on that paper, where they looked at the natural evolution of vaccinated people and they saw that it naturally automatically accounts for all the variants. Do you know what a paper I'm talking about, Vincent?
Yeah, that was I think Theodora and Paul, and I don't remember who else was
maybe
on
it was Michael Nusenswag and that collaboration.
Yeah.
But it was like a Nature paper about like a week and a half ago.
Yeah, I saw that. I don't remember who was on it.
Yeah.
Well, definitely Theodora and Paul's name came to the... Oh, there they are.
So the CureVac is an mRNA vaccine, but it doesn't have modified nucleotides.
Yeah. Right.
And the founder says you don't need them.
Well,
and he might,
I beg to differ.
I don't know.
I think the numbers beg to differ.
Yes.
I think the numbers beg to differ.
He says in a quote in this blog post, why would you need chemical modification? This makes sense for gene expression for proteins avoiding immune responses. In terms of vaccines, I'm not convinced to have these modifications. Well, maybe now you are. I don't know. So that you think that's a main reason why these didn't perform as well? They didn't make enough protein?
Yeah. Mm-hmm. Yeah, and we already have data from the Pfizer and Moderna vaccines in places with the variants showing that they seem to be just as effective. And so...
Are these lipid nanoparticle vaccines also the CureVac?
What is their delivery system?
I believe so.
They've got... There is something that's...
Well, there has to be. Otherwise
you just get nothing. Yeah.
Right. You'd have to electroporate into somebody's arm, and I don't think the electroporator is going to work that well. It is a lipid nanoparticle. So it really has to do with the fact that the RNA is just not modified in the appropriate fashion. And then you make very little of the protein so that you stimulate takes far longer to stimulate and get going an immune response, and therefore you get, or it gets cleared much faster, and therefore you don't have the right balance to overcome 47%. Although Vincent would argue 47% effectiveness of a vaccine is sufficient because it's about the rate of polio.
It's actually an interesting blog post. They go through the sequence, and it has, look at this, guys, it has a poly-C stretch. They added it.
They added a poly-C stretch, whereas we've taken it out of EMCV because we called it inhibitory.
So it says here, it's poly-C will bind PCBP and may enhance its stability. Well, if it does do that, it didn't help.
Right.
It also has a nucleus, a histone stem loop that can nucleate histones. I don't know why you'd want that, because this is an RNA vaccine, right?
Well, a nuclear stem loop, if it's in the RNA, enhances recruitment in a different fashion than the poly-A tail enhances recruitment at the ribosome.
Mm-hmm.
So it's not bound by the... so all histone mRNAs are not polyadenylated. They have this 3' stem loop. And that stem loop facilitates in a different fashion with different RNA binding proteins to facilitate and retain the ribosome.
So yes, these are lipid nanoparticle delivered, right?
So it most likely has to do with the stability of the RNA because it's not chemically modified, because histones are mass produced.
Yeah, but CureVax, I'm sure their stock is tanked, and they need something to blame.
How interesting that they would blame the variants. That's kind of unique, isn't it?
No, that is not at all
Never
unique.
would have heard
Always
of
blame the
such
variants.
a thing. Never heard such a thing. Really?
It's not because we
virus?
reopened everything. It's the variants. Yeah.
It's not, it's always the
It's
virus.
always the virus.
No?
Yeah.
All right. Alan, are you next?
I think I'm next. So I have Karen. Karen writes, "Hi, TWiV. Once again, thanks so much for keeping us all updated during the pandemic. More than a year after discovering TWiV, I still listen regularly. My basic science background, MD, MPH training decades ago, and 2 years of bench science as a research assistant before med school, doesn't always prepare me to completely grok the science you discuss, but that doesn't keep me from being amazed and so thankful for science and scientists. For instance, the segment on camelid antibodies was just remarkable. I'm sending you another episode from my daughter's YouTube channel, Science IRL, and provides a link. It's based on her interview with Dr. Vilain Motaze, head of the Notifiable Medical Conditions Program of the South African National Institute of Communicable Diseases, about his team's effort to contain spread from the first known case of SARS-CoV-2 infection in South Africa. I think Dr. Motaze's study is remarkable for at least 3 points. First, South Africa's approach to containment was based on team visits to the homes of contacts of the first identified case in South Africa, March 5th, 2020, for testing and for education concerning isolation to protect others from infection. It made me realize that I don't remember hearing about this basic public health tool being used in U.S. cities. Has the U.S. public health infrastructure just atrophied to the point where it is not feasible to employ this tool? That's a big part of it, yes, as a matter of fact.
Well, atrophy implies that it was ever present.
Oh, it's gotten booms of funding for some things. Post 9/11 and the anthrax attacks, we got a massive boost in public health spending that dried up, I think.
Doesn't mean it doesn't mean it went into this contact tracing.
No, it didn't necessarily go into that. And also it dried up in the next election cycle. Second, Dr. Motaze, who led the team into the field to visit people's homes, is a medical doctor and an epidemiologist. He did not send his team out to do this dangerous task. He led his team out. Excellent. 3, the team performed their duties with great consideration for the people they were visiting, even though this was a dangerous and complicated task that was sometimes carried out in the middle of the night. In order to avoid stigma for the household, they took the trouble to back their vehicle into the driveway and lifted the trunk to shield them donning their PPE and entering the house. Something about that just touched my heart. All right. Thank you, Karen. That's very
nice.
Cool.
John writes, we'll do one more round. Dear esteemed doctors, TWiV, 19C and sunny here in Greater Braddock, what I regard as like a perfect summer day in Stockholm. In about 24 minutes, I don't know what episode we're talking about. Well, maybe we could figure it out. You commented that the authors were epidemiologists, not evolutionary biologists or virologists. They're not protein chemists either. What I'd like to see added to a table of variants is which protein the substitution is located in, as well as what element of secondary structure in the native structure it's in: alpha helix, beta strand, reverse turn, or random coil. You commented that alanine to valine isn't much of a change. What was that? Yeah, I remember that. What episode? What alanine to valine? Oh, this is the...
this is from the variant paper, right? The last week?
I don't remember.
Is
Isn't much for changing. Chemically, it isn't, but valine is beta branched. Going back to the old Chow-Fasman tables, which I know have been improved on, but I don't know what's regarded as providing the best index of secondary structure potential these days. Alanine has high helical potential, while valine, with its pair of beta methyl groups, is disruptive to helices. So if the ALA in question is already in a beta strand, I suppose that a valine substitution would act to stabilize the sheet that it's part of. As far as which protein the substitution is in, it seems that the ones we hear about are in the spike. Oh, I think this was the RATG paper where they looked at binding to cells of RBD.
Oh, okay.
Is that because that's where we mostly look because of methodology? Thinking along those lines, too, if RATG13 is the closest relative currently known, I assume that the differences are spread across the entire genome. If so, wouldn't that also point to SARS-CoV-2 not being engineered, since something from a lab would presumably have tweaked the spike and left the parts more inert relative to infectivity alone? Probably this point has already been made. I just don't recall it specifically. Nomenclature. My first thought when the new Greek system came up was, what happens when we run out of Greek letters? How about using the same system as with hurricanes? Relatedly, naming vehicles after disasters and barren wildernesses, avalanche, tundra, doesn't seem to have had an adverse effect on sales in that sector. The cars called Avalanche and Tundra.
Oh, yeah.
Once they got going on that, they branched off into cults, Buick Enclave. I liked it better when people drove things like Packard Caribbeans, Constellations and Panamas, Kaiser Vagabonds, Ford Galaxies, and Pontiac Catalinas. On the other hand, in the 30s, there were a different series of Studebakers, President, Commander, and Dictator. Oops! Dictator was changed to Commander in 1937 for obvious reasons. Best regards, John. Oh, this is great. Yeah, car nomenclature. I don't know. I like the Yunnan, Y-R-A-T-G. Yunnan is Y-N, right?
Just to abbreviate.
Yeah, just two letters to
Right.
say where it came from. I think that the Greek is just information devoid, right?
Yeah.
Shouldn't a nomenclature have some information in it?
You know, there is a system
necessarily.
of alphanumeric characters defining grid squares all over the globe, called the Maidenhead system, because it was in Maidenhead, England. There was a conference where they came up with this, and these grid squares actually go down to very small squares of latitude and longitude. I live in FN 32. So you could have of that in the virus name. You could put the grid square.
Well, I believe that there's something similar because, like, when you do like Illumina sequencing, you're on a grid. Like the flow cell is all these grids because that's how you cluster, and they are different coordinates. And you can, and since if you know anything about Illumina, you know that the camera just takes a picture every cycle, or every time a nucleotide goes through, it takes another picture and looks for beacons of light. And that's how it aligns itself, is by the coordinates on that grid of where that beacon of light came. That's how it knows, you know, at position A336,000, B20, B22, you know, you put it in a C.
Right, but I'm just saying you could use the existing grid square system, the Maidenhead grid square system, as the location for the discovery of the virus.
I'm just supporting that we do that.
We do grid squares for other things too.
We do that when we collect sequencing data. and
And
stuff.
it wouldn't have the name of a
has
place
nothing.
in it, but it would be an alphanumeric that you could set in, and
Yeah.
it would give a very specific
Yeah.
bit of location information.
Yeah. But as we've said, just because you isolated it there,
No, it doesn't mean
means nothing
that
about
that's where it's
anything.
from, right?
So I'm not sure that where you've isolated it is really all that
Okay.
important. At the
I
end
think
of
it
the
is
day.
important, unless you know one way or the other. All right, let's put it this way: it could be, or it might not be important, right? So
Or
why
it might
throw
not
away
be.
the data,
Right, so-
just in case it could be?
Well, why? Well, then you should.
Well, you're not throwing
you
away
should
the
code-
data just by naming the virus.
Right, you're keeping the data. You're just not including.
You're just
it
not
in
mentioning
the
it in the name.
Yeah.
You have polio dot
No, I
P1
agree.
Mahoney.
Doesn't have.
alterations here, blah blah blah. Then you have polio dot P1 Bartholomew or whatever the one from the bee lady was or whatever it is. You have
Brunhilde.
these mutations.
Brunhilde.
Brunhilde. Okay.
No, I
Yeah,
agree.
well,
Those are not good.
I knew it
However,
was a-
Lansing is good. P2 Lansing, because it came from Lansing, Michigan. But I know we don't want to name places. So the problem is, if you say alpha, the site of origin of discovery is in a table, which you then have to find. And
But so is P1 Mahoney.
And so
It's
why
in the
not
table. P1 is in a
encode it,
table
either
where
as
you have
Alan
to
says
find it.
or with 2 letters. That's my proposal. That's it. We're not going to agree.
Because it stigmatizes people. And why do you want to stigmatize
Right, but if you
a group?
encode the location in a way that it's not obvious to the general public,
Yeah,
then
I
it's
think
fine.
so.
Could be fine. I mean, but
The other thing that occurred to me is you could just be very, very hyper specific about the location. So we wouldn't have Wuhan coronavirus; we would have fish market virus.
or you could even be more specific. You could say you have 1211 Cooper Road virus.
Right.
Now I'm going to get bombs in my mailbox.
You know, I mean, if you have no complaints, then 1211 Cooper Road virus is perfectly reasonable.
Make it specific
right?
and yet generic at the
Look,
same
look,
time.
Wuhan.
Well, it is.
And we have
I
done that.
didn't
We have
say
done
where
that,
it
for
was.
example, with Semliki forest virus, right? So
Yeah.
the story is that the virologists asked,
forest
we're
virus,
gonna name
right.
this after the place, and they asked the guide, "What do you call this place?" And the guide said, Semliki. Oh, okay, it's Semliki forest. Well, Semliki is actually the local word for forest.
Right.
So it's like we call it the forest.
Amy,
What are
can you
talking
take the next one?
Sure. Stephen writes, in the Atlantic, don't fall for these lab leak traps. Recent coverage of the pandemic's origin has ensnared readers in semantic quibble, side points, and distractions by Daniel Engber. And then he gives the link to the Atlantic article. Semantic quibbles, etc., sound like an okay topic to discuss, so I began to read. But I had to stop at this stunning false equivalency. The Columbia University microbiologist Vincent Racaniello said the same on his podcast this week in Virology. It's just crazy because there's no evidence for a lab leak. There's plenty of evidence for the natural origin. Others claim the exact opposite. There is still zero evidence to support the theory that the virus emerged from nature, Mark Thiessen, I think that's the way you pronounce his name, announced in the Washington Post, and mounting signs that it did not. According to Wikipedia, Thiessen is a graduate of the Taft School in 1985, a private prep school in Watertown, Connecticut. He earned a bachelor's of arts degree from Vassar College in 1989 and completed graduate studies at the Naval War College. I'll leave it open to TWiV to compare Vincent's bio in the relevant subjects. Note that he's a virologist. Stephen.
Yes.
This is a pet peeve of mine, and we get called arrogant all the time.
What is
Don't we,
is this a
Vincent?
false equivalency, Amy? Well,
What do
I'm
you
just
think?
asking.
I think, comparing a quote
Yeah.
about a virus's origin from a virologist with a quote from
for sure.
Bush 2's speechwriter, I think there is definitely a credibility gap between those two sources.
Well, that's right. But my response always is like, and I say this a lot. I say, Alan, you should feel offended also because we've wasted 6 years of our lives where we could have been drinking heavily, making a lot of money, going to parties, blah, blah, blah. Instead, we slugged it out in Vincent's lab. Oh my God, getting a degree that implies expertise in the field. And then I'm called arrogant because they're like, oh, she always goes back to the PhD. And I'm like, well, if you understood how hard it is to actually complete, you would be insulted by the fact that everybody negates it, right?
Because it doesn't help.
Right, but that has nothing to do with the actual that has nothing to do with being a graduate student and going to the pain of the bench work where you come to that pivotal moment in time. We have a special name for it where nothing works for months on end. Alan, you can relate to this. And you go, oh, really? I have no colonies?
They're going to call you arrogant, Amy.
Oh.
That morning where you wake up and you realize there is not necessarily a path forward.
Exactly. What do you mean morning? You imply one morning.
No, there was one. For me, there was
months of morning.
Any curve has a bottom, and I can actually remember waking up a particular morning and finally having it really sink in that this could just not work out.
Well, anyway, the point is that you can't counter a virologist's idea with a pundit. You have to have two virologists.
No.
Right.
And don't get David Relman because he's not a virologist.
At the very least. Right.
Right. And don't get the person who debated Daniel either because he's an anatomical pathologist.
Let's do one more, Alan, please.
Okay. So where are we? We're at Martin.
Yeah.
Okay, Martin writes, hi, TWiV team. You may not have heard this story from India, where an administrator is blaming a virulent new strain for his own failures. It was covered on the BBC News website and it's probably been covered elsewhere, provides the link. In short, these Muslim-inhabited islands, which I had never heard of, have a new administrator appointed by the Hindu extremist national government. Among other things, he has banned beef. He has also relaxed quarantine restrictions, so COVID cases went from 0 to quite a few. Figures are in the article. When queried on this, he blamed a return of economic activity. But first of all, in seventh to last paragraph of the BBC article, the virulent new strain. I suspect he is not one of a kind, sadly. No, he is not one of a kind. Martin is in Brussels. Thank you, Martin.
Yeah, I mean, he heard it on the news and he just repeats it. And then other people think it must be right if a leader of a country is repeating it. And there you go.
It's because of the virulent variant. It's not because of anything that I screwed up. Our vaccine would be great if it wasn't for those wascally viruses mutating.
Well, let me read this last one because it's actually a call to action and we should get this out. So this is from Allison. As a loyal TWiV listener, I hear a lot of references to scientists' past work with HIV. The term MSM, or men who have sex with men, is well known to all of you as a key demographic breakdown in HIV studies. Can you name a study that breaks down data by LGBTQ plus demographics? When it comes to SARS-CoV-2 COVID-19, we have almost no facts on the intersections of the pandemic with SOGI individuals and communities. SOGI stands for sexual orientation and gender identity. I'm writing to ask you to consider getting Microbe TV to sign onto an open letter to health leaders requesting a routine collection of SOGI data in healthcare settings. In short, the request is to have medical providers start collecting and reporting on SOGI data, the same as for other demographic data like gender. I hope you can see how this work would be a contributor to your unified call for proactive pandemic virus and public health preparedness. The National LGBTQI Plus Health Roundtable, the National LGBT Cancer Network, GLMI, formerly the Gay and Lesbian Medical Association, the Trevor Project, and more have joined together in signing this letter to raise awareness that, as Vincent would say, we just don't have the data. Organizational sign-ons to the letter would be low effort, no cost way to send your patients, students, listeners, colleagues the gift of data this Pride Month. Extremely appreciative in advance for your time and consideration about adding your organization, saying if you care about lives, collect the data. And there is a link. I unfortunately didn't have the link here. It got lost in the pasting, but I will put it back in so others can sign this too. It's a matter of going there and saying, I signed this. Yeah. Thank you, Allison. All right, let's do some picks. Amy, what do you have for us this week?
So I don't know. It's summer and I was in an ocean kind of mood. My family's at the beach. And so I was learning about how starfish actually have eyes. And then I saw this really cool article about a relic that's in the sea that looked really cool. And it's kind of like a sea pug or sea dog, like a sea pig or something. And it's in Catalonia. So I just thought it was
That's
really
cool.
cool.
These are
And it's from really old, from the Jurassic
brittle
era.
stars.
Yeah.
Neat.
And it's all because my family is at the beach this week and I'm here.
Very cool.
I just thought that they were kind of cool. And it was different. It was like non-virus related.
Very
Yes.
cool. Yeah, I like it.
So
Jurassic. Do we have any Borna endogenous element from the Jurassic? No, that's more than 100 million years ago, right?
We wouldn't know. It would be so far. Yeah, we can't get RNA from there. Or DNA
Well, from animals. Some mammals lived then, right? So we could do orthological stuff. But
we could have inherited it, but if it by now,
how long ago was the Jurassic?
Mutated.
Was when?
That predates.
It's 200 million years ago.
About.
So there have been some retroviruses dated to 400 million years ago. So it's possible that there's some Bornas, but they didn't have it in that database. Cool.
Yeah.
Alan, what do you have for us?
I have a book that I read a little while ago called World of Wonders by Amy, last name I'm going to butcher, Nejukumatathil. I apologize. She is actually a poet. I'm not a big reader of poetry, but this is a book of essays, regular prose essays, and has some lovely illustrations. Forgot the artist's name, but these are just short essays on her thoughts as a amateur naturalist, essentially, and it's just beautiful. It's just lyrically beautiful writing, and she's got really interesting insights that are, each chapter is based around a particular animal, and she writes about everything from the declining populations of fireflies and how our lights are messing up their sequence of flashes to more uplifting stuff about axolotls and just all sorts of really cool stuff. She's a big fan of corpse flowers, which I definitely appreciate too. And she weaves that into her own experience of growing up. She has a very, very interesting and unusual background for what she does, and it's just lovely essays, wonderful writing with a natural science theme. Very enjoyable. And they're short too, so you can read one of these in 15 minutes, and if you're in the middle of something, in between steps of your cooking or whatever. So just a wonderful little book to have around.
I was wondering where the name came from, so I looked her up. So she's American-born.
She's American-born, Philippine, and Indian.
Yeah, Malayali Indian.
Yeah,
very interesting. India. Kerala. Wow, it's an interesting name.
Nejukumatathil.
Wow.
Or something to that effect.
Cool,
and
very
if
cool.
she's listening, maybe you can write in and tell us how to pronounce your last name, but I loved your book.
Neato. All right, so I have, my pick is Tom Buck's YouTube channel. Tom Buck was a teacher. I mean, I discovered him early last year, and he was a digital arts teacher at a school, and he's teaching from home, and so he started doing videos on how he was using technology to teach, and his channel has gotten very popular, and I have a feeling he quit his job, but because he says he taught digital media for 11 years. It sounds like he's done, but Anyway, I don't know Tom, but he's got a nice channel, and he tells you how to do things, teaching and using equipment, and he's very engaging and does a good job. And I've learned quite a bit from him about how to use technology to improve communication in various ways. So,
Very cool.
Tom Buck, he has a lot of corny jokes, and it makes him kind of funny.
I approve.
We have a listener pick from Victor. It is sweaty hot in Bonn. I do not want to know the temperature. May I suggest a listener pick? It's a wonderful, entertaining, and well-sourced video by H. Bomber Guy on the Andrew Wakefield paper that claimed the MMR vaccine caused autism. I knew the MMR vaccine study that started the anti-vaxx movement was fraudulent, but I had not realized it was such a show. That's a famous, that's a phrase that Amy likes to use, right?
I prefer that or Orama. I actually prefer
Orama, S-Orama.
It's more encompassing.
Well, S-Orama is even worse than an S show, right?
Right.
Yes.
I was just reading an article on NPR. It was about the current state of the shipping industry, and they
which
used the
is
term, also
an S-Orama.
Right. No, they used the term clusterfest.
And
I thought that was hilarious, because that's what you do when you're writing for a family website and you want to use.
I was going to say, because usually that's not the term I've heard.
It's spelled a little differently when I use it, but I
that's another good one, Amy. We maybe want to use.
I don't use that one.
Why?
I don't use
it's too rude.
It's too rude, and it just this other one because when I grew up, we would say it was crapola on a stick.
Okay. Anyway, he didn't realize it was such an S show. The video is much too long for the internet age. So sad, isn't it?
So are ours.
But each time you think it cannot get worse, there is another pylon that demonstrates it is even worse. Okay, so highly recommended. He links to that. Thank you, Victor. It is a S *** show.
It is astonishing.
And it continues. Wakefield continues to this day, right?
Well, right. But I remember Brian Deere's original reporting on that. And as it was coming out, it was like, wait, I knew this was bad, but my gosh.
Yep.
Yes, but what tops it all off, which is the cherry on the top of this lovely Sunday, is the fact he lives in Texas and has put feelers out, and in previous years has run for House and Senate.
Right.
For governmental positions.
Yeah.
And he's gotten votes.
He's got money too.
Yeah.
It's very sad.
Which is...
Yep.
It's a cluster... What is the word?
Clusterfest.
Clusterfest.
The word. Well, yes.
Yeah, sure.
However,
I was thinking it was more flabbergasting.
TWiV is not a clusterfest. TWiV is good stuff. TWiV 771. Show notes, microbe.tv/TWiV. You can send us questions and comments to twiv@microbe.tv. If you enjoy what we do, consider supporting us. microbe.tv/contribute. Amy Rosenfeld is at Columbia University, enterovirus.net. Thank you, Amy.
Thank you.
Any plaque assays to do today?
Yes. This is interrupting my 3 trays per day rate.
I'm sorry. Well, we appreciate your sitting in.
Alan, did you do 3 trays a day?
3 trays. I don't remember how many trays I did.
Per day.
I did. There was a phase where I was doing very large numbers and stacking up the.
Yeah, but you probably cheated because you probably did 6 well plates.
I did 6 well plates. Yes.
No.
What are you doing?
Oh, you do the individuals.
We're doing individual 35 millimeter.
Ooh, hardcore. My big thing was the scintillation counter. I went through vast quantities of scintillation vials because I was for my thesis work.
I'm going through vast amounts of mice.
Okay.
Equally as dangerous.
Did a little bit of mouse work. Was not a big fan of that, but
I didn't say I was a fan.
No, when it's got to be done, it's there is that. Rush rather do plaque assays.
Alan Dove is at alandove.com. Alan Dove on Twitter. Thank you, Alan.
Thank you. Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.